Hello everyone, in this tutorial we're going to talk about transgenic plants and how do we insert novel genes or genes that are not usually present in plants inside plants in order to improve crop production or for various biotechnological purpose and so forth. So the rationale behind this plant's transgene uh, theory or approach is there is a transfer DNA or a tDNA that is usually taken from the TI or tumor inducing plasmid of a bacterium known as Agrobacterium tumefaciens. So TI tumor inducing plasmid. So this is a plasmid. You understand what is a plasmid? It's an extra circular piece of DNA that we find in bacteria. And for this case, our bacterium is Agrobacterium tumefaciens. Agro like agriculture, bacterium, this is a bacteria. Tumefaciens, it causes tumor. All right. So it's a tumor inducing, which means that it's a plasmid that induces tumor from agrobacterium. And the way it induces tumor is that it transfers a DNA from a bacteria into a plant here. And of course, the output is very ugly, not so good to see. You see roots or plants that usually carry this type of growth structure, which looks like tumor to me. And this comes from this bacteria because they are very opportunistic. They're found in soil, and this tumor, of course, is known as a crown gall disease in many dicotyledonous plant. As a result, once that tDNA enters, is very effective because it contains genes for plant hormones and opine production enzymes. These enzymes, as soon as plants start secreting those enzymes on the surface, it's going to actually provide food for those bacteria. But for it to work, it needs to find an opportunity, a point of entry. The plant tissue must be wounded in some point. So when scientists realize this, is like since this bacteria is actually inserting a plasmid inside a plant like this is a plant, and this plasmid contain a transfer DNA that is going to carry the gene of interest. What they've done now is that what if we intercept this process and we modify this plasmid? Instead of having this disease gene, we could now have a plasmid or a TI plasmid that will carry whatever gene of interest that we have inside this plant so that the plant could actually produce that. A good example is, I think, with tomato. You see that usually you can modify the genes such as leucoprene so that your tomato are not too ripe by the time they get to the supermarket and so forth. So for many biotechnological reason, you can actually intercept this property. Instead of putting the gene that may cause a disease or may cause this tumor, you can now insert your own gene of interest, and before you know it, you have a very nice transgenic plant. I actually found this very nice diagram adapted from a research paper from the journal Agrios in 1989, adapted by Aditya Anya. It explains the normal pathway. You're going to have plant infected, you have healthy plants and infected plant. The crown gall bacteria, what they do, they're just over there in the soil, and as soon as they make it there, you see this is the bacterium, and it's going to transfer the DNA here. If you notice, you see there's a transfer where the tDNA leaves the bacteria, enters the wounded plant cells. As soon as it enters, the tDNA integrates into the plant chromosome, and once it integrates into the plant chromosome, you have this rapid division of these transformed cells, and they keep growing and growing and growing. That's the reason why you actually get this crown gall disease. So now that you have this background information, we can actually jump into the three major steps that needs to be followed in order for us to have this transgenic plant. The first one is when you have the plant of interest, or the leaf at least, you need to cause a cell damage because the bacteria they need a point of entry. And as soon as the cells are damaged, it actually releases some hormones that are going to attract the bacteria to actually infect them. And you incubate those cell damage plant or leaf with agrobacterium that carries your disarmed TI vector. So your normal TI vector will carry, let's say, this red disease gene, and the disarm one here, they are going to carry this green nice gene. This is like the bad guy, this is like the good gene. So these are going to carry this disarm gene. Then your next step will be, now you move your disc into a shoot inducing media, so a media that is going to induce the formation of shoots. Of course, you will need to screen the tDNA markers as well. When you insert the plasmid, since you're already working with plasmid, you know how we do, you may add some antibiotic resistant gene in such a way that only cells, plant cells that have taken up this nice plasmid are going to grow. And eventually, after a couple of weeks, what you're going to have is that your shooted disc are going to be moved now into a root inducing media, and your plantlets can now be transplanted into soil, and you just wait for your very nice result there. All right, so this is the picture that represents it best when we look at it. And the approach that we're going to talk about now in detail is a tDNA mediated delivery. All right, let's just look at the literature here. What is happening on the side of the story. All right, so here's what we have. First, a disc is cut from, let's say, a tobacco leaf plant. This is the example that we're using now, and we culture them with Agrobacterium tumefaciens, carrying your recombinant TI plasmid that contained at this point. This plasmid is going to contain both a selectable marker and your transgene of interest. So if this is my transgene of interest, then I'm going to put my selectable marker as well here. And that's what you have here. You incubate them genetically, and once you've incubated them, this is what is going to happen on the inside. You're going to have you replace now the transferred DNA. Remember that the bad gene, that transferred DNA, is replaced with a selectable marker, kanamycin resistance, so which is a resistant gene that allows those cells to resist an antibiotic known as kanamycin. You're going to have some 25 base pair repeat region, and now you've put a new transgene of interest. Let's say you want to insert now luciferase. You've put luciferase now in the middle there. So that is the composition of your new tDNA. Some tDNA repeats, your transgene of interest, and a selectable marker. And of course, this tDNA is going to be here. Now, once you have it at this point, you select for plant cells that have actually taken up the recombinant plasmid that are going to be kanamycin resistant, and you grow them in a media that is going to resist, of course, kanamycin. Once you've grown them that way, the plants that are going to survive are those that you're going to take now into this, that are going to form a callus. Then from a callus you have what? A shoot inducing medium. Then you have a root inducing medium. Then you're going to have a plantlet with your roots, and you move it there as well. Also, what we need to mention, which is important, the damaged cell at the leaf disc edge, they actually release what I spoke about earlier, phenolic defensive chemicals. And these phenolic defensive chemicals, those are going to attract your other Agrobacterium tumefaciens to activate the virulent genes eventually. Then, of course, the encoded virulent gene, when you look at it, they're going to, of course, remove the TI plasmid DNA region, resulting in a T strand. What you see here, it's going to remove this tDNA and make it as a T strand here. Maybe I should go for maybe a red color here for you to see it properly, but you can feel free to zoom in and pause. It's going to have this T strand, and of course, eventually you're going to have a lot of virulence related protein that are going to create on the other side a T complex. So there's a T strand and there's a T complex now inside the plant cell. So inside the plant cell, you're going to have this T complex, and what they're going to do, they're going to interact with your T strand that you're trying to insert. And of course, this combination will actually direct everything at the nucleus, where your tDNA now it's going to bind to the genome and become integrated. So if you follow the trend from our plasmid, you have a T strand, then you have a couple of virulent protein that are actually carrying, like escorting, accompanying this tDNA as a complex, and until it reaches the nucleus. And once it's inside the nucleus, obviously it's going to become integrated there. And it's only the plant cells that have taken up the plasmid that will express now that selectable marker and expand into a callus. Then once we have the callus, ultimately you're going to have your shoots, and from shoots, roots, plantlet, and before you know it, you have a transgenic plant. I really hope that this short video was helpful. It will help you at least read further. It will always feel free to stop, rewind, and rewatch it as many times as you can. Thank you very much.